was here.
Yeah, it's definitely here, don't worry.
Okay, yeah.
Okay.
Thank you. Okay, hi everyone, my name is Tony. So today I'm presenting RAWG, which is an RNA-Seq workflow generator. So to start, I will give a quick overview on RNA-Seq analysis. RNA-Seq uses next-generation sequencing technique to study transcriptomic data. The aim is to compare the expression level between different contrast groups and identify the significantly differentially expressed features. There are many types of RNA-Seq analysis, so we mainly focus on differential gene analysis. And we think that a typical RNA-Seq pipeline can be broken down into 4 stages: alignment, assembly, analysis, and post-analysis. And those are all the tools currently RAWG has and all the interconnections between them. So file format is handled automatically, and all the intermediate and the utility steps are added by RAWG as well. So why is RAWG needed? Well, the graph on the left shows the RNA-Seq analysis result when different tools are used at different stages of the pipeline. And as you can see, there's quite a bit variation in it. So the number of significant genes found is different when different tools are used at different stages. And what's more important is if you imagine a Venn diagram, the different pipelines and the intersection is actually pretty small. So we think it's beneficial to compare the result from different analysis pipelines. So because RAWG focuses on a subfield of bioinformatics pipelines, it allows us to build a much more user-friendly interface. So users no longer need to use visual programming to define workflows like many platforms such as Galaxy. And also allows users to run an arbitrary number of user-defined workflows all within one session. So under the hood, RAWG uses common workflow language as the foundation. So CWL is widely used workflow standard. And by using CWL, workflows generated by RAWG can be readily reused on another platform. And CWL also brings portability and provenance to RAWG. So this is how we imagine a typical CWL analysis platform is set up. So on the bottom, you have the hardware level and all the software on top. So RAWG scope is highlighted in yellow. And in essence, RAWG contains a web portal, a workflow generator, and a set of RNA-Seq2 wrappers. Now I'll give a quick walkthrough on RAWG's functionality. So to create a session, user can select from a list of pre-indexed genomes or upload the genome files themselves. And an arbitrary number of conditions can be defined here. So if more than 2 conditions are supplied, RAWG will automatically run pairwise comparison for all the possible pairs. So samples can be added like this. Both pair-end and single samples are supported. And compressed files can be added as well. And the workflow is defined by select from a list of drop-down menus like this. As you can see, no visual programming is needed at this stage, which is the main feature differentiate us from other platforms. Finally, after submission, the job is added to the queue automatically, and this workflow schematic is generated as well. So for visualization purpose, and the CWL workflow file and the input file can be downloaded from this page as well. So in summary, RAWG makes between pipeline comparison easy, and it focuses on user-friendly interface. So there is also a possibility to extend to other fields by provide a different set of tool wrappers. So I would like to thank Derek for supervision, Suhail for sysadmin support. Thanks to Wellcome Trust for funding my study and this trip. Comments, questions, suggestions are welcome at our GitHub page. And we have a demo server only for the front end at this address. So my poster is still up at P40 if you haven't seen it yesterday. Thank you.